Hey, Mr. P here. Welcome to RNA Transcription. In this video, we're going to talk specifically about how DNA codes for RNA and then how that RNA is modified in order to produce the protein in translation. This first picture is a really good kind of summary of the processes we've talked about. You can see individual components of the entire IB syllabus. All the way back from the protein structure of topic 2, where we can see the beta sheets or the pleated sheets in this kind of purple region, where it was kind of the accordion structure, and you can see the alpha helical structure, the alpha helix in these corkscrew type structures. This is an example of a protein in the quaternary structure. Remember, it's a three-dimensional folded polypeptide. You can see individual amino acids. Amino acids are the monomer of the protein. And so we've got amino acids. We've got those amino acids folded into chains that make up a quaternary protein, in this case, a polypeptide. You can see from topic 1, cell biology, there are all kinds of cellular components. You've got the individual ribosomes, which are in charge of making the proteins. You have the mitochondria in charge of making ATP. You've got the nucleus over here that has the nuclear pores, and out of those nuclear pores you can see several molecules of RNA. That's the topic of this discussion. But inside of that nucleus we also have DNA, and we've talked in length about the structure of DNA, how DNA is folded, how it's coiled, why it's supercoiled, what holds it into the supercoil. You can see a replication bubble. In this case, it is a transcription bubble, or it is it's showcasing transcription. Okay, so transcription. But anyway, this lecture is all about RNA transcription. You can see that each of these ribosomes right here, this being ribosome, is all about making the proteins, but in order to get to the protein we have to make a molecule of RNA first. Okay, and so the purpose of this video is to talk about how RNA is made. First thing we need to talk about though is the central dogma of molecular biology. What is the central dogma? We've talked about replication, which is DNA becoming DNA, or DNA replicating itself, producing more copies of DNA. And so we know that in DNA replication, it is highly, there are a lot of enzymes that are in charge of DNA replication, but one of the most important enzymes is DNA polymerase, and specifically DNA polymerase 3. Remember, DNA polymerase 3 is the one that's actually going to synthesize the new strand of DNA in a 5' to 3' direction, reading the complementary template strand to produce a copy in a semi-conservative fashion. Remember, A pairs with T, C pairs with G. A pairs with T twice, those are the hydrogen bonds. C pairs with G 3 times, those are the hydrogen bonds. And we get a molecule that looks like this, where it is double-sided, okay, or double-stranded. One runs from left to right in this case, one runs from right to left, and so that's DNA. I'm not going to talk a lot about that because we're going to get into transcription. Replication being the first step of the central dogma, transcription being the second step. We are focusing on this step today. RNA polymerase. Notice DNA polymerase down here is the enzyme in charge of replication. RNA polymerase, just like DNA polymerase, is in charge of making the RNA molecule, but because we're not making DNA, we're not using DNA polymerase. We're making RNA, which can be seen here. RNA is a single-stranded molecule. It still runs in a 5' to 3' direction, just like DNA does. It's still going to be synthesized in a 5' to 3' direction, but we are not making a double-stranded molecule. We're making a single-stranded molecule, so we're using a little different enzyme. Also, we're using RNA nucleotides, and so you'll notice that U is seen in RNA, whereas T is seen in DNA. So there are a couple differences between DNA and RNA, but in order to make a strand of RNA, because it's different, we need to use a different enzyme. So RNA polymerase is going to make RNA molecules through the process of transcription. And then once we make the mRNA molecule, this being mRNA or messenger RNA, we're going to use that messenger RNA as a recipe in order to make protein. Protein is our end goal. We start with DNA going to protein. Protein is the way we express our genes and allow cells and organisms to do things with the DNA instructions that are held in the nucleus of every cell. Okay, the step 3 in the central dogma, again, DNA replication being the first step, DNA or RNA transcription being the second step. The third step is protein translation, and translation occurs in the cytoplasm, whereas RNA transcription and DNA replication all happens in the nucleus. Okay, so that's DNA replication, transcription, and then protein translation. This is the central dogma of molecular biology. This is really, really important in terms of our overall processes leading from DNA to protein. But like I said, we've gone through DNA replication in detail. Now we're going through transcription. The first thing we need to do, though, is talk about the types of RNA. So throughout this lecture and throughout the syllabus of IB biology, you'll notice that there are several types of RNA. One of them is mRNA. mRNA stands for messenger RNA. tRNA stands for transfer RNA. And rRNA stands for ribosomal RNA. So we've got 3 different types of RNA. You can see all 3 types in this slide. We've got our messenger RNA, which can be seen here. It is a single strand, 5' to 3' of RNA. Messenger RNA, just like it said down here, is our messenger, and so this would be our recipe that we use to produce a protein ultimately during translation. This big purple blob is our ribosomal RNA, so this would be our rRNA. And then over here you can see these kind of black squiggly holding an amino acid. This would be the tRNA. The purpose of tRNA is transfer RNA. It transfers the amino acid to our ribosome and ultimately builds the protein using the machinery of the ribosome in order to do that. But transfer RNA can be seen here. An example or a close-up look of the transfer RNA can be seen over here. This is a transfer RNA. It looks like a cloverleaf. It has that cloverleaf structure. It has an amino acid attached to the 3' end. It has an anticodon between the T loop and D loop. It is actually called the anticodon loop. This anticodon, GAA, is actually going to be complementary to our messenger RNA codon. The codon in this case would be CUU. Those would be the anticodon or the complement of the anticodon. This would be anticodon, GAA. This would be the codon on our mRNA strand. It would be CUU. The CUU is going to bond using hydrogen bonds to our anticodon. That's what actually links the tRNA. Again, anticodon right here. That's what actually links the tRNA to the mRNA to produce the protein. But that's going to be a topic in our next video, which is translation. But for now, I want to talk about what the 3 types of RNA are: mRNA, tRNA, rRNA. You'll see all 3. They are all made using DNA. They all have different functions. They're all structured different. It makes sense. Okay. Notice though that this tRNA molecule, while it looks 3-dimensional and it looks like it's a weird kind of cloverleaf design or cloverleaf shape, it still has a 5' end and a 3' end. So you can actually see that the 5' to 3' is actually going to go this way, loop around, loop around, loop around, and end at the 3' end. So this is still a single-stranded molecule. It actually just kind of folds on itself and produces these hydrogen bonds to form these loops, which give tRNA the cloverleaf design. But it's really important that it folds over like that. Notice that the G and C still bond 3 times, and the U and A still bond twice. So you're going to see that over and over, whether we're talking about DNA replication or RNA transcription. But this design is really, really important when it comes to actually fitting into not only the ribosome, but actually attaching itself to the messenger RNA codon in order to link these amino acids to a grain of, or a chain of amino acids, a growing chain of amino acids to produce the protein. Okay, but this is translation. We will talk about that later. Okay, let's talk a little bit more about transcription. Transcription is, like we said before, is DNA turning into RNA. So DNA is double-stranded, RNA is single-stranded. We are going to specifically turn DNA into a strand of mRNA, the messenger RNA being probably the most commonly known type of mRNA or RNA. But this process occurs in the nucleus. It is going to use DNA, this being our double strand of DNA, it's going to use DNA in the nucleus to produce a strand of messenger RNA, which will travel out through the nuclear pore of the nucleus into the cytoplasm, where it will meet with a ribosome in order to produce a protein. Okay, there are, like we said before, 3 types of RNA: messenger RNA, tRNA or transfer RNA, and rRNA, ribosomal RNA. And there are 3 steps of transcription. There is initiation, step 1, elongation, step 2, termination, step 3. We can see all 3 steps in the next few slides, but in order for transcription to take place, there has to be a promoter region on our DNA. And again, all of these regions are actually on the DNA. Notice there is no actual mRNA made yet. You have in yellow right here the gene. That is what is going to actually produce the protein. We know from genetics that genes code for, they're the coding regions of DNA. They code for a specific protein. That protein is obviously going to be expressed in order to produce a certain phenotypic object, whether it be a physical trait or a physical characteristic. And then you have a terminator. Terminator is going to be the stop signal. Promoter is going to be the start signal. And between a start signal and a stop signal, you have a gene, which is the regulatory section that codes for a protein. Okay, start site, stop site. DNA, remember, has an anti-parallel nature, and so this bottom strand is going to go 5' to 3' right to left. The top is going to go 5' to 3' left to right. And so we have 2 strands, but we're only going to synthesize one of them or utilize one of them for a template strand. And it is RNA polymerase that is actually going to begin the synthesis of that RNA molecule at the promoter region. Remember, promoter region is our start site. But in order for RNA polymerase to build the RNA molecule, it has to actually determine which strand is going to be the template strand. Because we have 2 strands to pick from, we are always going to produce RNA in a 5' to 3' direction, just like we did with DNA. And so if we look at this bottom strand being 5' to 3' right to left, that's going to be our template strand. If we're going from this start site to stop site, we know that we have to transcribe this RNA molecule from left to right, moving from the start site to the stop site. And so we can't use this top strand going left to right, or we would be making our new 5' to 3' RNA strand in the exact same direction as our top strand. It would be 5' to 3'. So we would be basically using the DNA strand this direction and building an RNA strand in the same direction. That is not anti-parallel, so that is not correct. We cannot use this top strand. We have to use the bottom strand, and so in order to do that, we have to, like I said, determine the strand. So the top strand is said to be the sense strand, and the bottom strand is said to be the anti-sense strand in this example. Now, not all top strands are the sense strand, and not all bottom strands are anti-sense strand. Anti-sense strand is the strand that is actually going to be used as the template, and the sense strand is the one that is not the template. Okay, and so we are always going to transcribe the anti-sense strand. In this case, in this picture, the anti-sense strand is the bottom strand, and so that's really, really important to determine, first of all, before we can look at how initiation elongation, and termination happens. We have to determine which strand is going to be transcribed. In this case, it is the bottom strand. In other cases, it might be the top strand. Okay, but notice we have the antisense strand. It is the strand that is actually going to be transcribed. The RNA polymerase, which is this big molecule that is blue, RNA polymerase, is going to start at our promoter region, which is the start signal, and it's going to work in a 5' to 3' direction to our terminator. This would be the stop site. And so once the RNA polymerase gets to the terminator, it's going to disassociate, or it's going to fall off. It's going to be prompted to quit, and we have a molecule of mRNA that is made, but it hasn't been modified yet. And so we call that molecule a pre-mRNA molecule because as we will go into later on in the video, it has not been modified. It has not been the introns have not been removed, the exons have not been spliced together, it hasn't been, or the poly A tail methyl G cap has not been added, and so those are things that are vital in order for this mRNA message to withstand the pressures of the cytoplasm once it's removed from the nucleus. Okay, so let's talk about the specifics of initiation. There are several things that have to happen in order for initiation to take place and therefore transcription to progress. One of the main things we talked about so far is the promoter region. Promoter region, remember, is the start signal for transcription, but transcription can't happen at the promoter without a certain series of steps that have to take place. Okay, first of all, these activator molecules have to attach themselves to the enhancer region of the DNA. Again, this strand is the DNA. This is not the RNA yet. This is the DNA strand. We are talking about how this DNA strand becomes readable and how transcription can take place, how RNA polymerase can open up the DNA molecule and read the template strand, again the antisense strand, to produce the pre-mRNA or the eventual RNA molecule that will leave the nucleus and then therefore produce a protein. Okay, so the activators bind themselves to the enhancer region. Notice that this enhancer region is upstream of the promoter region, but there is often quite a bit of distance between this enhancer region and the promoter region, and so it requires a DNA binding protein. This DNA binding protein does exactly what it sounds like it does. It bends the protein or bends the DNA molecule and actually places this enhancer region right next to or extremely close distance-wise to the promoter region. Notice that this is quite a bit of distance. However, when you bend the DNA here and fold the enhancer region back towards the promoter, you can essentially bind to the enhancer region and the promoter at the same time. This would be the beginning of this RNA polymerase initiation protein complex. Okay, and so notice that from this single strand of DNA that has an activator region or an enhancer region and a promoter region that are several units in distance away from each other, you fold it with the use of DNA binding protein, and now you can make this entire protein complex bond to everything at the same time. We have RNA polymerase, which can be seen as this kind of translucent or translucent white enzyme. This is the molecule that will actually produce the RNA molecule. It is going to synthesize the RNA in a 5' to 3' direction, like we said most or a lot of times. And this entire complex is actually going to help initiate. Notice that this is called transcription initiation, or the initiation phase of transcription. RNA polymerase is the enzyme that is responsible for transcribing the strand of DNA and producing the RNA, but RNA polymerase can't do it alone. It takes this entire initiation complex, and it requires not only the binding protein, but a lot of mediator proteins and activators in order to activate the process and get it to start. Okay, so that is initiation. If we talk about initiation in terms of what are the steps that you would want to include when you answer a transcription question. The first 3 marks would probably come from a series of statements that look similar to these. Activators and transcription factors bind to the enhancer, which is here, and the promoter, which is here, region of DNA, which is going to increase the rate of transcription. So we have activators that are going to activate the rate of transcription. We have RNA polymerase and a bunch of mediator or transcription factor proteins that are going to not only bind to the enhancer region, but are also going to bind to the promoter region, and they are solely responsible for increasing the rate. Notice we haven't talked at all about RNA polymerase yet, because RNA polymerase, as it says in statement 2, RNA polymerase binds to the promoter sequence, which is here, and is going to eventually separate or unwind the DNA strand and synthesize a complementary mRNA molecule from the antisense strand in a 5' to 3' direction. Okay, so we've got activators that are going to, and transcription factors, activators here, transcription factors here. They're going to bind not only to the enhancer region, but the promoter region of the DNA molecule, giving RNA polymerase the place to bind in order for RNA polymerase to synthesize that molecule of mRNA in a 5' to 3' direction. Okay, that is initiation. So what is elongation? Elongation is the elongating of this mRNA molecule. Notice it started at 5'. It is synthesizing. This end would be the 3' end. It is being made this way using RNA polymerase. And we are reading the antisense strand. So this would be the antisense strand of this DNA molecule. This would be the sense strand of the DNA molecule. Notice this is the template strand. This would be the coding strand. These would be all of the transcription factors that are sitting at the promoter region where transcription, which is this entire process, would have started. And they would be moving, again, building this RNA molecule in a 5' to 3' direction all the way downstream until it reached a terminator. Okay, terminator would be the stop site. RNA polymerase moves along the antisense strand in a 5' to 3' direction, building that strand one nucleotide at a time. It is placing the nucleoside triphosphates. They are RNA nucleotides, but they have 3 phosphates. You can see what that nucleoside triphosphate looks like. It actually uses an enzyme, RNA polymerase, to take 2 phosphates off. So it's going to remove the 2 phosphates. You can see them here. And the enzyme is going to release energy while it removes those 2 phosphates. And so that energy is responsible for linking this nucleotide to the 3' end of this growing RNA molecule. Okay, so if this is our 3' end, this is our 5' end, it is making the RNA in a 5 to 3', using the energy associated with the removal of the 2 phosphates of this nucleoside triphosphate RNA nucleotide. And so we are linking those one by one in a 5' to 3' direction as we go from promoter to terminator. Okay, nucleoside triphosphates are added using the energy associated with the removal of the 2 phosphates. All RNA polymerase is doing is removing the 2 phosphates. By removing the 2 phosphates, you are releasing energy. The energy is going to successfully link this nucleotide to the 3' end, growing this chain by 1 nucleotide as it goes. Okay, so that's elongation. Termination. We have reached the terminator region of this DNA molecule. And as soon as we read or reach the terminator region, this RNA polymerase is going to disassociate or pop off. And once it disassociates or pops off, it basically terminates the production of this mRNA molecule. And so, if I can back up, this mRNA molecule, again, is not modified yet. It's a pre-mRNA molecule. We're going to talk about the mRNA and how we're going to modify it in probably the next video, because this video is getting a little long. But I want to go through, or I wanted to go through this process really quickly, but also slowly, in order for you to fully understand transcription from an initiation, elongation, and termination standpoint. RNA polymerase is going to move down this mRNA molecule, or this DNA molecule, until that terminator signal is reached. RNA polymerase is going to detach from the template molecule, or the template strand. We call that the antisense strand. This strand would be the sense strand. And as soon as the RNA polymerase pops off or detaches, the DNA is going to rewind. It's going to zip back up. And one thing that's really important to know is that there are many mRNA molecules that can be produced at the same time, meaning there are tons of RNA polymerases that can be synthesizing the same gene essentially at the same time. They just kind of follow each other. And that allows for multiple proteins to be produced from the same gene, especially if it's a gene that is utilized frequently in terms of the protein product that it makes. Introns, though, have to be removed, and the mature mRNA has to be made. That's called mRNA processing. We will talk about that in a later video, or in the next video. Okay, but for now, we've gone through transcription from an initiation, elongation, and termination standpoint. If you have questions, make sure you bring those to class. The very last thing I want to talk about is the transcription of multiple genes. You see from this video, or from this slide, that we have gene 1 that is going to produce a protein product, and we have gene 2 which is going to produce a protein product. Because this is a eukaryotic chromosome, or a eukaryotic section of DNA, we can actually produce multiple mRNAs from the same section of DNA, especially if they are different genes, because this RNA polymerase can actually be moving this way, coding this gene, or producing the RNA associated with the gene, and this RNA polymerase can actually be moving this way on the same section of DNA, as we make two different mRNA molecules from two different genes to produce two different protein products. Okay, the next video is going to be RNA processing, but for now, bring your questions. We'll talk about it in class. See ya.